How tobacco and cannabis use impact male fertility and genetic integrity, a hidden reproductive crisis. A recent study published in Scientific Reports investigates the detrimental effects of tobacco and cannabis smoking on male fertility, focusing on sperm quality, DNA integrity, and genetic variations in mitochondrial Cytochrome C Oxidase (COX) genes. The study suggests that cannabis may cause even greater harm to male reproductive health than tobacco. Infertility affects approximately 14% of couples globally, with male factors contributing to nearly 50% of cases. Despite the growing awareness of this issue, the rise in recreational cannabis use and the persistence of tobacco consumption remain significant public health concerns. Tobacco smoke is a well-known source of over 7,000 chemicals, many of which are carcinogens and mutagens, severely affecting sperm DNA. On the other hand, cannabis, particularly the active compound tetrahydrocannabinol, THC, has been found to disrupt sperm motility, viability, and morphology. Moreover, studies have suggested that cannabis use could alter the epigenetic landscape of sperm, possibly affecting future generations. As cannabis use becomes more widespread and legal in many regions, understanding its reproductive implications is critical. Further research is needed to clarify the genetic and epigenetic impacts of these substances on male fertility. About the study. This study examined semen samples from 113 men, categorized into three groups: non-smokers, NS, N=37, tobacco smokers, TS=39, and cannabis smokers, CS, N=37. Participants with alcohol dependency, chronic diseases, or genetic abnormalities were excluded. Semen parameters, including volume, sperm concentration, morphology, and motility, were analyzed following the World Health Organization (WHO) guidelines. DNA integrity was assessed using acridine orange (AO) and chromomycin A3 (CMA3) staining. Sperm samples underwent density gradient centrifugation to remove somatic cells before DNA extraction. Mitochondrial DNA (mtDNA) was isolated, amplified, and analyzed for genetic variations in the MTCO1, MTCO2, and MTCO3 genes. PCR primers were designed based on National Center for Biotechnology Information (NCBI) reference sequences, and sequencing was performed using the Sanger method. Fluorescence microscopy was employed to assess sperm chromatin packaging and DNA fragmentation. The study revealed significant findings about sperm quality and DNA integrity. Sperm morphology. Both TS and CS groups showed a significant decline in normal sperm morphology compared to NS 2.1. Cannabis smokers exhibited significantly lower morphology scores than tobacco smokers, p=0.002. Sperm motility. Progressive sperm motility was lower in the CS group, 10.18-10.6%, compared to TS, 13.1-10.6%, and NS, 14.2-11.3%, though the difference was not statistically significant, p=0.223. Non-progressive motility was significantly lower in CS, 20.63 plus 12.6%, compared to TS, 27.82, 16.6%, and NS 34.4 is 14.3, p.1.11. The immotile sperm percentage was highest in CS, 68.666, 21.9, 24.4%, and NS, 51.73, 13.8, and 0.01. DNA integrity. AO staining revealed significantly higher DNA fragmentation in CS, 28.53, 15.8%, compared to NS, 10.1, 14%, and TS, 6.4, 10.2, CMA3 staining indicated increased protamine deficiency in CS, 37.1%, compared to TS, 25.3, 14.9, and NS, 15.0, 15.4%. Genetic variations in mitochondrial DNA. The analysis of mtDNA sequences revealed 23 single nucleotide polymorphisms in MTCO1, 15 in MTCO2, and 30 in MTCO3. However, no significant differences in SNP distributions were observed among the groups. MTCO1, p=0.1, MTCO2, p=0.23, MTCO3, p=0.07. Both tobacco and cannabis use negatively impact sperm quality, but cannabis appears to cause more significant damage to DNA integrity. The elevated DNA fragmentation and protamine deficiency in cannabis smokers suggest a higher risk of infertility, lower embryo viability, and reduced pregnancy success rates. These findings highlight a potential hidden reproductive crisis, especially as cannabis use becomes more socially accepted and legalized in many regions. Reduced sperm health due to cannabis and tobacco use can lead to infertility, higher miscarriage rates, and long-term health risks for offspring. Public health campaigns should focus on educating young men about the long-term risks of smoking, both tobacco and cannabis, on fertility. Awareness and preventive measures could mitigate the growing public health issue. Policymakers and healthcare professionals should consider these implications when making decisions regarding the legalization of cannabis and tobacco consumption. Amor, H. Ismail A. Jankowski, PM et al., 2025. Effects of marijuana and tobacco on male fertility and their relationship to genetic variation of mitochondrial cytochrome C Oxidase genes. Scientific reports /doi.org/10.38/S41598-025-918940. Sharma R. 2020. Effect of tobacco smoking on sperm quality and DNA integrity in men. Human Reproduction Update 26-1, 65-77. Ganan C. et al., 2019. The Impact of Cannabis on Male Reproductive Health, a Comprehensive Review. Journal of Clinical Medicine, 8-1166. To doi.org, 10.339 org, JCM 81166.